What I will talk about this morning is just to provide some historical context for what's happened in biotech, and particularly in Cambridge and Kendall Square. And then I will make a transition to talk about convergence, which is something that this building has been dedicated to since we established the Koch about 5 years ago. So it's molecular biology to Kendall Square and convergence. And I want you to think back to when all of this began. I can't do that. Molecular biology, yes please, I would like that. Molecular biology really had its origins in the discovery of Watson and Crick in 1953. Yeah. And that discovery showed us for the first time how information was transmitted between generations. Before that, we knew about genes and chromosomes, but we did not know the chemical structure and the nature of the information that allowed traits to be transferred from one generation to another. And Watson and Crick made that discovery only a short 50 or 60 years ago. So really all of the contemporary molecular biology, cell biology, and research that we're engaged in across Kendall Square had its origins 53 years ago. And I'll remind you that Harvard recruited Jim Watson to start the department or division of molecular biology in the late 50s. And MIT recruited Watson's mentor, Salvador Luria, you've just heard commented on, to start the molecular biochemical department here at MIT. So here two universities really establish, just for the curiosity of understanding how biological systems work, the approach of a gene-oriented biology here in Cambridge. And that seeded the developments we're talking about today. It took about 10 years to go from the discovery of the structure of DNA, which is shown at the top here, to how information in that DNA flowed into the cell. From DNA into RNA, which is copied from the DNA, has a similar chemical structure, and then that's decoded to make the proteins that carry out most of the functions of the cell. It was shown by Jacob Monod, who got a Nobel for this work, that there were regulatory proteins that would bind to the DNA and regulate the flow of this information into the cell. So not only could you have genetic material, but that genetic material could be used in different ways in different cells at different times. Now, when I entered molecular biology, and in fact, when I came to MIT in 1974 to the Cancer Center, where we were going to understand the nature of cancer and develop cures for cancer, this is the way we understood the structure of our genes. When someone said they had a gene of a certain function, we thought about it in this context. That's because we had been studying the structure of genes in bacteria. But it became clear in the 70s, in fact 1977, that our gene structure was very different. Gene structure in our cells look like this. And what do I mean by that? You see these segments. This is a representation of the DNA in one of your chromosomes. And these black boxes are pieces of genes. And you will note that they're not contiguous. They're interrupted by pieces of non-information called introns. These are called exons. And these are spliced together to make the sequences that are then translated through that machinery to make protein. So our genes are fundamentally different. They have this novel structure. They have mutations that affect the splicing process that joins the information together. They are regulated in a very different way, a much more complicated way. You'll hear more about that today. And we didn't understand this existed as a gene structure until '77. We're still struggling with understanding how these genes vary in their expression from cell to cell. Because in one cell, you can have this same gene produced as a protein by splicing these two pieces together, but in another cell, splice this away to make a different protein. And until we understand this variation at a cell-by-cell basis, we will have a limited understanding of how cells work. And that's really where we're at in part of molecular biology. But then going further, in the mid-'70s, these processes were discovered out of the pure curiosity of trying to understand how a gene functioned in cells. We developed the technology to carry out DNA recombination, the DNA sequencing, and DNA synthesis. This is Wally Gilbert at Harvard and Fred Sanger. This is actually Gobin Khorana, who was recruited to MIT in the early '70s and synthesized the first gene and laid all the chemistry for the whole biotech industry. And this is Boyer and Cohen and Paul Berg on the West Coast and recombinant DNA. But Cambridge was seeded for this activity because in the late '50s, Watson was recruited to Harvard, Luria was recruited to MIT, and molecular biology began in this community. And that led to a lot of events in which the benefits of recombinant DNA were debated. And I'm going to play you about a 2-minute video of Mayor Bellucci opening the city council on the debate about recombinant DNA. And it's interesting in the context of the questions being asked and how that set the
Can you
table
make an
for
absolute
what we've done.
100% certain guarantee that there is no possible risk which might arise from this experimentation? Is there zero risk of danger? Answer that question later too, please. Would recombinant DNA experiments be safer if they were done in a maximum security lab, a P4 lab, in an isolated, non-populated area of the country? Question. Would this be safer than using a P3 lab in one of the most densely populated cities in the nation? Question. Is it true that in the history of science, mistakes have been known to happen? Question. Do scientists ever exercise poor judgment? Question. Do they ever have accidents? Question. Do you possess enough foresight and wisdom to decide which direction the future of mankind should take? Question. The great war poet Joyce Kilmer once wrote, "Poems are made by fools like me, but only God can make a tree." I have made references to Frankenstein over the past week, and some people think this is all a big joke. That was my way of describing what happens when genes are put together in a new way. This is a deadly serious matter, sir, madam, sir. Harvard University, this is a serious matter. It is not a laughing matter, please believe me. It is not a laughing matter, and this is for the National Institute. This is not a laughing matter. If worse comes to worse, we could have a major disaster on our hands. I guarantee everyone in this room that if that happens, no one will be laughing then.
So this was after the National Institute of Health had passed their guidelines, and we were debating in Cambridge whether these experiments should be permitted at MIT and Harvard, and under what conditions we should go forward with these experiments. And we met with the city council and answered those questions as best we could. We never said there could not be an accident. We never said we had absolute understanding of everything. We said We will do this in a responsible way. You can come into our laboratories and see how we're doing it. We will tell you what we're doing before we do it. And we gain the confidence of Cambridge that these experiments can be done in a safe way. And shortly thereafter, spreading off the West Coast, the first biotech company was Genentech, started by Bob Swanson and Herb Boyer in 1976. In 1978, Wally Gilbert, who's right here, and myself, this is Bernard Mock and Charles Weissman from Zurich, this is Ken Murray from England, Hans Schaller from Germany, an international group of scientists started Biogen, initially located in Geneva, Switzerland, and then shortly afterwards opened the lab here in Cambridge. And when we opened the lab in Cambridge, the mayor came and cut the ribbon and said, as long as you do the experiments by the same guidelines that the universities have, you're welcome to be in Cambridge. Because we had debated it, he had the confidence of the community that they were comfortable with this. And by the way, you pay taxes. Now, that's important. Because when I was talking about Kendall Square a few weeks ago, the vice mayor of Cambridge was there and said, biotech real estate taxes in Cambridge contributes $50 million a year to the budget of Cambridge. And with that, they built 3 new schools. They've expanded the teaching population. They're converting Cambridge to a city with really great infrastructure. So things have changed, and this is one step along that way, and that was the ground foundation for why biotech, in part, came here to Cambridge. Now, in addition to the research in the mid-70s and 80s on recombinant DNA, a second revolution happened in Cambridge, and that was the genomic revolution, really led by Eric Lander, who's now the director of the Broad Institute across the street, which is an institute shared between MIT and Harvard. Eric contributed more sequences to the human genome sequence than any other laboratory. It was clearly an international effort, but it has brought genomics to Cambridge in a way that provides the basis for big data and the engagement of molecular biology on an international biomedical integrated fashion. And in late 203, 50 years after the discovery of the structure of DNA, we obtained as a community the ability to sequence the human genome. We finished the human genome. But more importantly, we developed the ability to actually sequence almost any organism on Earth. The microbiomes that occupy you, the plants that are around you, the birds that fly over you. Every creature will be sequenced. And all those genes will go into databases, and we will learn biology at a level that we've never had before. And you will realize that if you put together the chemical synthesis of genes, the ability to synthesize organisms, and the ability to learn the complexity of all the genetic makeup of organisms, that is the future of what molecular biology and modern biology and life science is. And it's going to provide food, it's going to help us understand the environment, it's going to provide energy, and it's going to provide better health across society. It also has led to this. And this is the Kendall Square schematic of Kendall Square. You see here in these yellow buildings along Charles, MIT. And you see in blue, biotech. This is not all the biotech organizations in Cambridge, but the major ones. And over here, there's Novartis, International Research R&D Center. Pfizer here, Sanofi there. So large pharmaceuticals are moving in and establishing research laboratories here in Cambridge. And that trend is continuing and in fact strengthening. Biogen occupies this set of buildings over here. Amgen is over there. Alnylam, a company I'll comment about in a few moments, is down here on 3rd Street. This is a bed of innovation. It is supported by MIT here, Harvard up here, Mass General across the river, Harvard Medical School across the river, BU Tufts, and all the intellectual capacity this beautiful region has. It's just remarkable what's the richness of high tech in the biological sphere and in other spheres has in this community. And this is changing the country, the world, and certainly Massachusetts. But this is where we're at, and we're moving forward in this whole field of biotech and biological research. This represents Cambridge. This is the nickel factory over on Mass Ave with Nico and its beautiful flavors, and I love those as kids. And now we got DNA painted around the same stack. So Cambridge has undergone a transformation of a remarkable type that has changed our community in much of the country. Now that change is still ongoing, and this is one of the most interesting things I think that's happening in Cambridge. This is Google, who just opened one of its major research and development sites here in Cambridge. Amazon is in Cambridge, IBM is in Cambridge, Alchemide is in Cambridge. IT to health, IT to biomedical research, IT to communications with patients and controlling the flow of patients is an integral part of how engineering and IT and information technology is going to be able to make medicine more efficient, more controllable, and more predictable. So this part of the community, I think, is a major part of what's going to happen in Cambridge over the next 10 and 20 years, in addition to the ongoing innovation that you're going to hear about in biomedical science. So Cambridge is in a position to really move forward and bring Massachusetts and Boston and the rest with it, in my opinion. Now, not to give you the idea that all innovation and all discovery in life science is complete. We know everything. We know the human genome. By the way, all we have to do is crank all those DNA sequences out and we'll understand biology. Quite the contrary. And I'll use an example of how this discovery to innovation is something that's still ongoing in biological science. The fellow on the left there is Andy Fire. Andy was a graduate student in my lab here at MIT in the Cancer Center. He showed up in the early 80s. He had a hairdo like this. He was 19 years old. He had graduated from Berkeley as a math major. And he wanted to study biology. So in the Department of Biology, we really don't care how much biology they know. We can teach them biology, right? What we can't do is teach smarts. And he had a very good background in mathematics, and he had taken biochemistry and genetics, and he was an extraordinarily talented student. Went off to Cambridge to do a postdoc with Sidney Brenner, who also got a Nobel Prize, by the way, and then went down to the Carnegie Institute in Washington. When he was there, he collaborated with Craig Mello, who's out at U of Mass, brilliant guy, and the two of them discovered RNAi. This was in about 2000. We didn't know about this whole process before 2000. We didn't know that little bits of RNA is made in every one of our cells and controls genes. Not just one gene, it controls almost all of them. And we didn't understand, we didn't even know it existed until 2000. So in 2000, with the discovery of RNAi, we did some biochemistry at MIT and came to the conclusion that I have to say, I have every conflict of interest in what I'm going to say in the next few minutes, you can imagine, all right? I'm a co-founder, I'm chair of the scientific board, I'm a member of the board of directors, and I hold equity. So I can't have more conflicts than this. All right. But we developed some biochemistry here, Tom Tushel, Phil Zamor, David Bartel, and developed some intellectual property, and then working with venture capital, started Alnylam in 2002. We really didn't understand the generality and the specifics of the process in our cells, but we knew that if we could take little bits of RNA, which we could use in the lab called small interfering RNAs, and introduce them into cells, they would silence a gene in a gene-specific way. I could design, knowing the DNA sequence, this little RNA and silence a gene. So instead of a drug development program that might take 5 years, I can do a drug development program in 3 months. So this is, give me a gene, give me a disease, can I treat that disease? So we started Alnylam in 2002, and what we've come to as a method to actually use these small RNAs is outlined here, and it's targeted specifically to the liver, and it's targeted to this receptor on the surface of liver cells, the acyl-glycoprotein receptor, and it binds at high affinity with a form of GalNAc, a hydrocarbon that's on the surface of many proteins. Those proteins bind to it, this receptor, get internalized in these little vesicles within cells, and then get recycled back to the surface. And what we've done is to take these small RNAs, tag them to a carbohydrate in a specific way, get them taken up into the cell. They leak, I think, out of these vesicles because we really don't know the process by which these vesicles work. In fact, we don't know how many actual pathways of vesicles there are in cells. It's still an area that's not well explored, but fundamentally important in terms of the delivery of drugs to cells. And so we can deliver these RNAs with reasonable efficiency to the liver of patients. So how well does that work? Well, I'm not going to talk about the biochemistry, but you need to do the chemistry. But I just want to give you an update. This is public data over the last year or so in clinical trials. This protein here, PCSK9, controls cholesterol in your system. It's a protein produced by the liver, circulates in the blood, controls your cholesterol level. And if you use an siRNA and you give it in, this is human data, 1 injection. This is a placebo, water. This is 100 milligrams, this is 300 milligrams. And you see 100, you get partial reduction, and 300, you get a reduction to 80%. And the striking thing is 1 shot sub-Q in your fat pad, and I have a lot of fat pads, so I don't have to worry about this. You get over 6 months the silencing of that gene. If I have another protein, this is C5, it controls a complement, and it controls a depletion of platelet in many disease processes. You can inject 1 dose, and this goes over 3 months. You can get silencing. So give me a gene, I can make an RNA, I can modify that RNA, and I can inject it, and I can silence that gene. So we can do this in liver, and there's many different clinical trials ongoing. I can't tell you how beneficial it's going to be. We'll have to open the envelope and look. But it is a whole new modality to treat disease. And if we can push this on down to another order of magnitude of efficiency, I think we can take it to most tissues in the body. So I think it's a whole new era of a new modality, much like monoclonal antibodies and protein factors in the treatment of disease. But the general point is, still novel discoveries are becoming available to be translated into science. And the last one is CRISPR, is the development of a whole novel engineering tool to genetically manipulate cells in tissue in vivo. We don't know what it means. We just discovered it a couple of years ago. It's going to take us 10 years, maybe 15, to figure all of this out. So we've gone through the discovery of the structure of DNA in the 50s, molecular biology in the 70s with recombinant DNA developing, genomics in 2000 through 2000, and where are we at now? So we've called this coming 20 years. I want you to remember this. I think it's very important. Universities have the responsibility of looking 20 years ahead and saying, what's going to be important? It's not tomorrow. It's not next year, 5 years. We need to develop the future. And that, in a time scale in science, is 10 and 20 years at a minimum. So we made commitments there to do molecular biology. We made commitments in recombinant DNA to do biotech and into the genomics. And now, what are we making commitments to? Well, this building is an example. And that's convergence, the convergence of engineering with life science. So convergence is the blueprint for innovation, advances in information technology, material, imaging, nano, optics, quantum physics, coupled with advance in computing, modeling, and simulation, have already transformed the physical world. And that's this device and the things like it, right? It's really fundamentally changed our lives. But engineering combined with life science will transform life science, biomedical science. And it's something the country needs to invest in, and we've been investing in it in terms of MIT, in terms of the Koch. So this is where you're sitting in the Koch Institute, and in this building there's 12 engineers and 12 cancer biologists, and we're intermingled on every floor. There's not a structure in the country like this. And we're dedicated to cancer research. We do some other things because it all mixes together and advances our understanding of biological systems and our ability to translate. And you'll hear from many of these people. Tyler Jacks is our fearless leader. He's a magnificent leader and scientist. Daryl Irving here is an immunologist who's in engineering, in chemical engineering. Susan Hockfield is in the building, a previous president. She's taken an office here. Michael Seema is an engineer that does implanted sensors, among other things. Angie Belcher uses phage to make imaging devices of small tumor sites in the body. Dane Winthrop is another engineer who's an immunologist. Bob Langer, you'll probably hear about later. So it's a mixture of engineering and biology, bringing the tools of engineering, the skills of nano, the skills of micro fabrication into biological science and activities. This is an example. This is from Daryl Irving and Dane Winthrop. One of the most exciting new developments in cancer treatment is immunotherapy, is directing the immune system to reject a cancer. And what they're doing is expanding that science, meaning we know this is reactive, immune system is effective on lung cancer, melanoma, and some bladder and some colon. They all have high mutation loads. Neoantigens is what they're called. And the body has an immune response to them and has a checkpoint that the tumor uses to resist the immune system. And that's these checkpoint antibodies here. And what they're doing is using ways of delivering novel vaccine peptides with lipids attached to them, so they'll go in the blood to be taken up by these cells, boosting the immune system with some IL-2, this is Dane Winthrop's technology, and then analyzing or activating macrophage with antibodies to specific cell surface proteins on these cells, tumor cells, and boosting and directing that immunotherapy to different tumors. And as the last example, so this is in mice, and They can show that if you have untreated progressing over 6 weeks, and then that's the spread of melanoma in these animals, and treat them with that cocktail, and you see almost complete suppression of that malignancy. So it's steroids. It's immunotherapy on steroids for a specific subset of patients. Now, do we know this is going to work in patients? No. It's got to be translated. It's got to be taken in clinical trials. It's got to be tweaked and worked with clinicians, and that's why we have relationships with Dana-Farber and Mass General and other clinical centers around the country to take this type of engineering and technology to its impact on patients. So what I've tried to give you is an oversight of where biotech came from, where engineering integration into life science is. A third of all MIT engineers now, a third are involved in life science. A significant fraction of our physics faculty are involved in life science. A significant fraction of our mathematics, applied math, is involved in life science now. MIT is a really hotbed of the engagement of modern IT, mathematics, algorithms, big data, and molecular and cell biology in biomedical research as part of this community that spans from one end of the city to the other. Thank you for your attention.